When you think about the most complex instruction manual ever written, what comes to mind? A spaceship? A supercomputer? Well, the real answer is actually inside every single one of us. So today, we're going to decode the blueprint of life itself, our genetic code. It all kicks off right here. A single moment, a single cell, formed when an egg and a sperm fuse together. This one tiny cell, called a zygote, somehow contains the entire set of instructions to build a whole human being. It's mind-boggling, right? So how are those instructions written? How are they organized? And how in the world are they read? To figure that out, we need to open the book of life. Okay, so imagine your entire genetic code is like a massive, ancient library. It's got every single piece of information needed to build you and keep you running. But, to find anything in a library that huge, you need it organized into volumes. And in genetics, these volumes are what we call chromosomes. And here, we get a peek inside that library. What you're looking at are called karyotypes. They're basically a photographic inventory of a person's chromosomes, all neatly paired up and arranged by size. Now, we humans have 23 pairs. The first 22 pairs are called autosomes, and they carry the instructions for pretty much everything, eye color, height, you name it. But it's that final 23rd pair that holds the key to biological sex. You can see on the left in the male karyotype, we've got an X and a Y chromosome. And on the right, the female karyotype has 2 X chromosomes. So there it is, the complete library, all 46 volumes, ready to go. So if every single one of our cells needs a copy of this library, how does the body go about making new additions? Well, it turns out there are 2 incredibly important but also very different copying processes that our cells use to pass on all this genetic information. All right, let's break this down. First up, we've got mitosis. The easiest way to think about this is as the body's perfect photocopier. It's what we use for growth, for repair. You get a cut, mitosis kicks in. It creates 2 new cells that are absolutely identical to the original, each with the full library of 46 chromosomes. But then there's meiosis, and this is a much more creative process. Its only job is to make reproductive cells, sperm and eggs. Instead of making a perfect copy, it creates 4 unique cells. And here's the most important part. Each of these cells only has half the library, just 23 single chromosomes. Why? So that when a sperm and an egg get together, they can combine their half libraries to create one brand new, complete set for a totally new person. The whole process is really elegant, but the real magic, the thing that makes each of us one of a kind, happens in step 2, crossing over. The chromosome pairs, one you got from your mom, one from your dad, they line up next to each other and literally swap sections of DNA. This step is absolutely crucial. It's like taking 2 different decks of cards, shuffling them together, and then dealing out brand new, unique hands. This genetic shuffle creates totally new combinations of genes. After that, the cell divides twice, and the result is 4 sperm or egg cells that are completely unique. It's the very reason why you and your siblings, even with the same parents, can look so different from one another. But as you can imagine, a process that involves copying and dividing trillions of bits of information is incredibly delicate. And sometimes, glitches happen. Errors can creep into the system, and that can lead to what we call genetic abnormalities. We can basically group these errors into 2 main buckets. First, you can have mistakes that involve whole chromosomes, entire volumes from our library. This happens when meiosis doesn't divide the chromosomes up perfectly, and a sperm or egg cell ends up with too many or too few. The second kind of error is within a specific gene. Think of this like a tiny typo in a single sentence in one book in that whole massive library. The book is there, but a key word is misspelled, and that changes its entire meaning. So, what happens when these kinds of errors actually occur? Let's open up the case files and take a look at some of the most well-known conditions caused by errors with whole chromosomes. The most common one is Down syndrome, which is also known as trisomy 21. Now, trisomy sounds complicated, but it literally just means 3 bodies instead of the usual 2. In this case, a person has an extra copy of the 21st chromosome. So instead of a pair, they have 3. This happens in about 1 out of every 700 births, and it results in a distinct set of physical characteristics along with disabilities in intellectual and motor development. Now, these errors can also happen with the sex chromosomes. Take Klinefelter syndrome, which affects about 1 in 600 males. Instead of the typical XY pairing for a male, these individuals have an extra X chromosome, making their genetic makeup XXY. This can lead to certain physical traits like being taller than average and can sometimes result in impairments in language or other academic skills.
And on the flip side, we have Turner syndrome. This affects females who are actually missing an X chromosome, so their genotype, their specific genetic code, is written as XO instead of the typical XX. This occurs in about 1 in every 2,500 female births. And what's really interesting here is that while individuals with Turner syndrome might struggle with math, their verbal skills are often quite good. It just goes to show how complex the effects of a single chromosome can be.
Now, Fragile X syndrome is a little different. It's not about the number of chromosomes, but about a defect on the X chromosome itself. There's an abnormality that causes a part of the chromosome to become constricted and fragile to the point where it can even break off. And because males only have that one X chromosome, they tend to be affected more often and more severely. This can lead to intellectual disabilities, a short attention span, and some distinct facial characteristics. Okay, so we've seen the impact of errors at the volume level with whole chromosomes. Now let's zoom way in. We're going to shift our focus to those smaller but just as impactful typos that can happen within a single gene. A classic example here is phenylketonuria, or PKU for short. This is caused by a recessive gene. The easiest way to think about that is you need 2 typos, one from each parent, for the condition to actually show up. If you only get one, you're just a carrier. Now, with PKU, this genetic typo means the body can't make an enzyme it needs to break down an amino acid called phenylalanine. If that stuff builds up, it can cause severe intellectual disability. But, and this is the crucial part, so hang on to this thought, this is totally treatable with a special diet. We'll come back to why that's so important.
Next up is sickle cell anemia. This is another disorder caused by a recessive gene, and it's most common in people of African descent. It causes red blood cells, which are normally nice and round, to form a rigid sickle or crescent shape. But here's where things get really fascinating from an evolutionary standpoint. Having just one copy of the sickle cell gene, being a carrier, actually gives you significant protection against malaria. But if you inherit 2 copies, you get the full-blown disorder, where these sickled cells can't carry oxygen well and they clog up blood vessels, causing a lot of pain and organ damage. Tay-Sachs disease is a particularly devastating gene-linked disorder. It's also recessive, and it's most common among Jews of Eastern European descent. A defective gene means the body can't create an enzyme to break down fatty substances, or lipids. So these lipids build up to toxic levels in the child's brain and spinal cord. This causes a relentless degeneration of the central nervous system, and it's tragically fatal by about age 5.
And finally, let's look at spina bifida. This is what's called a neural tube disorder. Super early in a pregnancy, this structure is supposed to fold over and close up to become the baby's brain and spinal cord. But sometimes, because of a really complex mix of genetic and environmental factors, it doesn't close all the way. This is another key example where nurture comes into play. We know for a fact that environmental factors, like the mother not having enough folic acid in her diet, can seriously increase the risk. You know, looking at all these cases brings us to a much, much bigger question. If our genes are our blueprint, does that mean they're also our destiny? Well, this is where the story gets really, really interesting. Let's go back to PKU for a second. Remember that one? The condition is 100% genetic. That's nature. But its most devastating effects can be completely prevented by a simple environmental change, a special diet, and that's nurture. This is a perfect, powerful demonstration of the dance between our heredity and our environment. It shows that the exact same genetic blueprint can lead to totally different outcomes. Our genes might write the first draft of our story, but they definitely don't write the ending. They're a set of possibilities, not a fixed destiny. And that brings us right to our final thought. With modern technology, we're getting incredibly close to being able to read our entire genetic code, every last letter. We can spot predispositions to disorders and understand our own biology in stunning detail. But all this knowledge comes with some heavy choices and some big ethical questions. So, knowing what you know now, I'll leave you with this. Would you want to read your own blueprint?